Hello, I'm Alberto Bassett. I'm the president of the Ecological European Federation, and it's a great honor and pleasure for me to introduce Professor Carlos Herrera, who is going to give our plenary lecture, and is the Haeckel Prize of the Ecological European Federation for 2017. So Carlos Herrera has got his degree and PhD in biology at the University of Seville, and he has entered in 1979 the Consejo Superior de Investigación Científica as a permanent scientist of the biological station at Doñana, where he has been professor of research since 1986. While he originally has been trained as an ornithologist, then his scientific interest and work have been subsequently focused on many aspects of the evolutionary ecology of the interaction between plants and animals, and these studies have actually contributed to deepen our knowledge on plant frugivore, plant pollinator, plant herbivore system, as well as on more complex antitrophic system. And Carlos Herrera has also given his attention throughout his career to the scientific divulgation, raising the public awareness on the value of biodiversity and healthy ecosystem, which I think is really an overwhelming topic, because actually we need to increase the ecological culture in Europe if we want to give value at the social and political level, even to our research and achievement. And another important point on the same field is that he was actually having a transfer of knowledge to conservation and ecosystem management that was managing to have a better management, a better conservation, the protected area where he's actually done most of his research activity. And as regards the last point, just a personal note, since his colleague and friends at Doñana told me that Carlos Herrera has dedicated his life and work at nature conservation, since she actually loves nature, loves to work in the field, make trekking in nature, observe animal and plant, and it is probably this interest on nature that has given to him the capacity to see and to understand before others the evolutionary relevance of what we can call as hidden characteristic of plant and animals, such as the sub-individual variation on which he is going to speak today in his lecture, and that letting him to open new field of research in the science on ecology. So I am happy to give the floor to Carlos Herrera for his plenary, and I also need to thank him a lot for managing to be here with us, actually traveling across many borders in order to reach us today, and this is a welcome because he's just arrived. So.
Is there something to change this? Is it forward? Is it forward? Okay. Well, thank you very much. It was not easy to get here. I arrived only 4 hours ago. So I hope my brain is functional now. Well, one of the missions of ecology is to describe and understand patterns and processes. Patterns and processes take place at a variety of spatial scale. Over the history of ecology, there has been a diversification of the science of ecology into different subdisciplines. And curiously, each of these disciplines is associated with one particular spatial scale. At the top of the gradient, we have macroecology. The mission of macroecology is to understand patterns at a very large spatial scale. Down in the scale, we have landscape ecology or ecosystem ecology, where we are working at the scale of 10,000 meters or so. And even lower, we have community ecology. and population ecology. Curiously, all these subdisciplines have in common one feature, and this is that the elemental unit is the individual. In the case of plants, it's the individuality of plants, made it very easy to understand what is a shrub, a tree, and so on. But there is still very low, in this scale, a narrow range of about 1 meter or 1 meter, 5 meters or so. This is what happened inside individual plant. There is no name for this kind of ecology. This unnamed ecology is what I'm gonna talk about today. It's what happened inside individual and what are the implications of sub-individual variation in plants. I'm making no claim for novelty. This is not new. It's been a long time now that plants are actually a collection of entities. 200 years ago, Charles Darwin's grandfather, Erasmus Darwin, was already aware that plants are a collection. He called a family or swarm of bats. This idea more recently has been expressed in more modern ecological terms, and plants have been considered a meta-population. So my approach will be much more simpler, even naive. My approach had to do with a very simple property of plants. Plants are made up of a repetition of homologous organs that perform the same function. Flowers, fruit, seeds, leaves, and my premise, a very simple premise, is that the considerable number of homologous structures produced by one individual plant are not identical. This is a very simple idea. It's nothing about the population dynamic of modules or something like that. It's about differences between modules. Okay? This is a outline. First, I'm going to talk briefly about what traits are changing within plants and how large is variation. Next, I'm going to show what ecological consequence has this variation in terms of spatial pattern, and then start the fun. What are the consequences? Is this important for the ecology of the plant themselves or for the animal consumer that eat the plant and lay it on the plant, pollinate the flower, and so on? Then evolutionary implication comes, and finally, the mechanism. Which are the mechanisms underlying sub-individual variation in plants? That's about genetics and epigenetics. And finally, I'm going to present a summary and provide some ideas about the future. What should we do to understand better not only the mechanism, but also the consequence in ecological and also in evolutionary terms? So let's see what is variable within plants. Well, to make a long answer short, everything is variable. Every trait you consider in sufficient detail is variable within plants. For example, everybody knows that not all leaves in a tree or in a shrub or in an herb are identical. If you take a sample of leaves, a sufficiently large sample of tree in a tree crown, you find things like that. Leaves are different in terms of shape or size, but also in more subtle parameters, like nitrogen content, water content, or even secondary compound composition, secondary compound concentration. Okay? But not only leaves, even flowers. We tend to consider flower as one of the most constant organs in plants. But flowers in the same plant or even in the same inflorescence are a variable too. Well, you can see the flowers, some small or larger, but if you measure nectar volume or nectar concentration or nectar composition, everything is variable between one flower and the other in the same inflorescence in the same plant. not to bore you, fruits and seeds are also variable. Not all fruits produced by the same individual are identical. Not all seeds produced by the same individual are identical. We trade variety, chemical composition, nutrients, secondary compounds, seed size, nutrients in the site, and so on. So, well, you can tell me, well, it's not so rare. There is variation. All right, but the key point is not that there is variation. The key point is that the variation is very large, much larger than we usually acknowledge. And a simple way to evaluate the magnitude of sub-individual variation is to make a variance partitioning. You have the whole population-wide variance in a given trait, say leaf length, or say fruit diameter, or say nectar concentration, and you have the population-wide variance, and you can make a partition into two components, the within individual and the between individual. I have summarized here a collection of investigation for flower, fruit, leaves, seed traits. I don't know if this is, well. The blue line indicates that 50%, all the points above the line indicate that variance within plant is higher than variance between plants. Except in flower, fruit, leaves, and seed traits have more variance inside the individual. The variance within individual is larger than the variance between individual. This has a number of consequences. The first of all consequences is that we are missing an important component of phenotypic variance in population if we ignore what is happening inside the plant. This is the first conclusion. And the second one is that if there is so much variance within individual, we are doing a poor sampling if we are not considering a good sampling design inside plant, okay? But at any rate, in any case, what we have is an interesting ecological phenomenon. Because if the variance within plant was very low, there is no room to speak further about that. But I think there is sufficient room for investigating further the phenomenon. The first thing to talk about is what are the consequences in terms of spatial organization of ecological parameters, say nitrogen concentration in leaves or water concentration in leaves or seed secondary compounds, things like that. Well, in some cases, what sub-individual variation is producing a gradient, a very clear gradient. This has been known for a very long time in the case of leaves. Most of you know that leaves in the top position in the crown of tree are different from leaves at the bottom position of the crown of tree. So there is a vertical gradient. It's related to light gradient, or there are other gradients like compass orientation. This example is water concentration in leaves of Prunus mahalet at the bottom and the top position in the crown. But this kind of gradient relative to some external reference axis are not the only one. There are also some smaller scale gradient taking place along axis of the plant. This is one example from the stinking hellebore, Helleborus fetidus. It's a comparison of what happened with the stoma in the lower and the upper position along the same stem of the plant. There is a clear gradient in the stoma size and stoma density running from bottom to top position. Yet another example of a small scale gradient is on flowers. In linear inflorescences, it's very frequent to find that nectar composition or nectar concentration in sugar is changing linearly along the inflorescence, like here, in this plant, it's a Gladiolus illyricus, and nectar sugar concentration declines from the bottom to the top position. So within individual variation has organization. Has organization means that there is a predictive power of considering the position. It's a good predictor of what's happening at the point when you are taking the measurement in relation to other points. But in any case, what is really important, regardless of what the mechanism, regardless of the gradient is broad scale gradient on a tree or a smaller gradient along a plant axis, the main consequence is that within individual change the graininess, the granularity of the environment. Consider the situation in which every tree in a population has all its organs identical. Or in other words, the variance within individual is near zero. In this situation, we had a coarse-grained environment. But we saw 2 or 3 slides back that this is not a real situation. The real situation is more like this, in which variance within trees or shrubs, in any trait you are considering, variance within individual is very high, sometimes is approaching unity. Most of the variance is within the plant. So finally, what we have is a fine-grained environment. This is a variogram. From the perspective of description of the plant community in functional terms, this scenario is completely different. From the perspective of animal consumer, these scenarios are completely different. So the first consequence in ecological terms of sub-individual variation is that it changes the granularity of the environment. But there is another important consequence. So far, has been considered sub-individual variance as a constant. But things are not that simple, as always. Different individuals have different level of sub-individual variation. So across space, there are individuals with high variance, and there are individuals with low variance. This I called some years ago the Haldane conjecture, because it was Haldane, the first one to say that individual plants not only have a characteristic mean, they also have a characteristic standard deviation. This was written many years ago, but apparently was unnoticed by most plant ecologists. What this means? Consider one plant, and you can measure sub-individual variability, for instance, with the coefficient of variation. In this case, this is a frequency distribution of the coefficient of variation of seed size, seed mass, in 100 plants of a daffodil. If you plot the frequency distribution of the sub-individual variation, you find that to the left of the graph, you have very constant plants. Some individuals are very, very similar seeds. At the opposite extreme, you have individual plants with very variable seeds. So there is actually individual variation in the level of variability. And the consequence is that the spatial distribution of local variance is also variable. One more example, the frequency distribution of coefficient of variation within plant, within stinking hellebore plant, more than 100 plants, is the coefficient of variation within plant of the stomatal index and the stomatal density, the stomal length, and specific leaf area. All these traits have important functional consequences. All these are important for photosynthetic performance, for water economy. Individual plants are different in their degrees of variability. Okay? This is it. I think this is suggesting some new question. This new question, what are the ecological effects? We already know that practically every trait you sample within plant is variable is very variable within plant. Individuals are not identical in the degree of variability. So are there some consequence? Is there some predictive power in the sub-individual variation level? The answer is yes. This is a simple example. We saw that individual stinking hellebore plants are different in their degree of variability. So it's very easy to relate the level of variability, the sub-individual variability, with fecundity. This is a very simple path diagram, and I have removed all this to avoid cluttering, avoid all the U arrows and things like that. One important conclusion is that sub-individual variability in important functional trait has predictive power for plant fecundity. The higher sub-individual variability in stomatal index, stomatal length, specifically area, and the lower the stomatal density, the higher is the plant fecundity. These four variables explain about 13% of observed variance across individual in fecundity. That's not a minor point, because individual difference in fecundity are important in not only in ecological term, but also in evolutionary term, because if you consider fecundity as a surrogate for fitness, there are differences in fitness related to differences in sub-individual variation. But even more interesting is the finding that differences in variability across individuals produce differences in fecundity. The interesting point is that different populations of the same species have different level of sub-individual variation, and these differences explain differences between populations in fecundity. So we have, to the left of the graph, population of stinking hellebore that are very constant from top to bottom position of the leaves on the same axis, and to the right population that are very variable for one of the traits, it's a specific leaf area. So two things. First, individual differences in variability has consequence in terms of fecundity. And second, population differences in sub-individual variability have consequence for fecundity too. But more generally, what sub-individual variability produces is conferring to the plants the ability to broaden the range of resources to be exploited. This resource, in these two graphs, to the left is a situation in which there is no sub-individual variation, so variance within is 0. To the right is a situation in which the variance within plant is nearly 1. So most variance take place within plant. Along a resource gradient, it can be abiotic, like light intensity or germination microsite, but also biotic, pollinator side, disperser side. What plants achieve is a kind of division of labor. Division of labor by broadening the gradient of environment it can exploit. It doesn't matter if the environment is pollinator size or is light intensity. So an example is provided by seed dispersers of one Mediterranean tree, this Philyrea latifolia. Philyrea latifolia produce fruit that are dispersed by several passerine birds. And the fruits are very variable in size. This is representative of one tree, and you have small and medium size and large size fruit. And these are the three main seed dispersers of Philyrea latifolia: European robin, blackcap, and the blackbird. Curiously, they select different sizes of seeds. The robin eats the lower sized fruit, and the blackcap eats the intermediate fruit, and the robin feed on the larger fruit. So we are producing a broader range of fruit in a tree, in the same tree. A tree is able to attract different size of bird dispersers, and in this way, they are broadening the ecological condition in which they sit are dispersed. So far, I've been talking about the consequence for the plants, but what about the animal? But the first consequence for animal is that sub-individual variation provides an opportunity for selection, for discrimination. When one animal comes to a plant, say to lay eggs, to feed on a flower, to feed on fruit, or something like that, the animal finds a variety of possibilities, of variants, among which to select. So the first consequence is that provides opportunity for selection. This is one example from the hawthorn, Cratigus monogyna. The Cratigus monogyna tree produces thousands of fruit, are mainly dispersed by thrushes, blackbirds, and so on. I wanted to know if after arriving to a tree, to a fruiting tree, the birds were selective or not. It was not difficult, because thrushes pick the fruit, touch with the bill, and drop it or not. So you can compare the fruit lying on the ground beneath the tree with the available fruit. In the graph to the right, every point is a tree, and the horizontal axis is the mean diameter of available fruit, and the vertical axis is the mean diameter of rejected fruit. So within each hawthorn tree, thrushes are selecting fruits depending on size, and they are dropping to the ground the largest fruit and ingesting the smaller fruit. So the conclusion is that after arriving to a plant with variable organs, animals have the ability to discriminate, and they do it. But there is no free lunch. There is another side of the coin. And the other side of the coin is that discrimination implies a cost in terms of time, in terms of energy. A very good idea, my friend. Okay, better now. Anybody that at some time has watched pollinator come into a flower, now that not all flowers are visited. Some, about 25 or 35 or 40% of flowers are approached but not visited. This is in this picture, there is nectar only in the white flower. I know it, and the bee now too. She has to select from different flowers with different reward, and it takes time. It takes energy in flight. So finally, the ability to discriminate within the plant has the cost in terms of time and energy, and finally will produce some risk-averse behavior. Two examples of risk aversion one by pollinator. This one is a Viola casorlensis, an endemic violet from southeastern Spain, with a very long spur, is pollinated by hot moth. Plants of Viola are different in their degree of variability of the spur length. To the left, we have the less variable plant. To the right, the most variable plant. It's very easy to see that fruit set decline with increasing variability within the plant. This is just what could be predicted from risk-averse behavior. So, pollinators are somehow discriminating against the most variable individuals are discriminated in favor of the least variable individual. A similar example, but this time from a non-mutualistic interactor. For a beetle, it's a seed predator or a tropical tree, Guattuma ulmifolia, the guattimo. They prefer to select the larger fruit, but they try to avoid those trees with the most variable fruit, again, supporting the idea that there is a risk-averse behavior. On the horizontal axis, we have a coefficient of variation of fruit length within tree. Every symbol is one tree. And on the vertical axis, we have the proportion of fruits on the tree that are predated. Again, we found support for the hypothesis that when faced with variable resources within a plant, animals tend to behave in a risk-averse way. So we have all elements now to start to talk about evolutionary possibility. First, we know that variation is very high. Then we know that not all individuals are identical in their degrees of variability. Then we know that there are reasons to speak that some individuals can be favored depending on the degree of variability, and other individuals are discriminated against. So we have the opportunity for selection. We have reasons to believe that sub-individual variation has some evolutionary implication. So in general, for many years, this has been the standard phenotypic selection equation in which we regress some fitness surrogate, fecundity, or some measurement of success of photosynthetic performance, something like that, and relate to a number of variables, the means, a number of variables describing the phenotype. But what about the variance? The variance is missing from this equation. So I proposed some time ago that it's very simple to expand the phenotypic selection model to incorporate the variance. So the only thing we need to do is to incorporate a set of variance terms. In this way, we will have the selection coefficient for the mean and selection coefficient for the variance. And the important thing is that for the same reason that incorporating different parameters, different means in the traditional equation, we are controlling for the correlation between measurements, we are controlling for the correlation between the means and the variance too here. There are not many data in the literature, including my own work, but the few data indicate that that's true. There is selection on the mean. We knew that for 25 years back or so, or more. But there is also selection on the variance. And the axes are the standard selection coefficient on some trait. Horizontal axis is selection coefficient on the mean. The vertical axis is selection coefficient on the variance. For only 7 investigations on flowers, fruits, and seed. There are very few studies with this information, but even these few studies are telling us that we need to consider the two possibilities at the same time, selection on the mean and selection on the variance. Because the mechanism, we already know what is the mechanism. The mechanism is animals selecting among plants with different degrees of variability, or the environment selecting differently on different individuals with different variability. Next comes the most difficult part of this problem, are mechanism. Mechanism, I'm not saying ecological mechanism. We will never understand what is happening with some individual variation until we know what are the real mechanisms underlying it. We know really little about this. We know very, very little. But I'm going to show you what little we know. At least, I think this data may suggest some experiment or some future investigation. Well, the first thing is that individual differences in variability has some genetic basis. Individuals that are different in variability are also different genetically. There are several different ways to test this idea. One is by means of classical quantitative genetic crosses and things like that. But what we did, what's a different approach, is using genomic scan. In genomic scan, you look for a large number of the genetic marker and look for correlation between individual genetic marker and the trait you are interested in. We screened many AFLP markers, anonymous genetic markers, and looked for correlation between individual variability. In this case, it's in the stomatal index, but we did with every leaf trait we were considering and looking for correlation. Remember, not all individuals are similar in their degree of variability. In this graph, we show that some particular AFLP markers are significantly related with the degree of variability. Yes, correlation between genetic marker and one phenotypic trait is not a proof of causality, yes. But at least it provides a working hypothesis to work further on this. At the very least, it provides a suggestion that individual differences in variability has a genetic basis. Remember, there's what variation, there's what selection, and now we found that there is also some connection with genetic. So there could be some evolution of the sub-individual variability level. But things, it seems to me that things are a bit more complex than this. I think epigenetics is involved too in sub-individual variation. This is the current definition of epigenetics. This is a bit different from the definition of 50 years ago by Conrad Waddington. So it's always useful to put the current definition. When we talk about epigenetics, we talk about heritable changes in phenotype or gene expression that are not encoded in DNA sequence. Okay? There are maybe cytosine methylation, there can be a histone modification, anything else. But the key point is that epigenetic differences between individuals are not connected directly to DNA sequencing, what we call usually genetic. The important point is that epigenetic change are able to produce phenotypic variants even if no DNA sequencing variation exists. So what happened with sub-individual variation? Some time ago, I hypothesized that even if plants, individual plants, have the same genotype all over the plant, if this genotype is the same, they could be epigenetic mosaics. And they could be epigenetically different in different places of the plant. And my first test was with holly, Ilex acifolium. You know, holly has spiny leaves, that the holly is producing spiny leaves in response to herbivory by mammals, and non-spiny leaves. We compare the epigenetic profiles of spiny leaves and nearby non-spiny leaves, and what we found was quite interesting. The probability of cytosine methylation of anonymous CCGG loci was different for prickly and non-prickly leaves, even if the leaves were close together on the same branch. On the same branch, a prickly leaf has a lower methylation probability, or in other words, there were epigenetic differences between spiny and non-spiny leaves, or in other words, holy trees are an epigenetic mosaic, and the epigenetic mosaics in holy tree matches phenotypic mosaic. So this was the first evidence we found of epigenetic mosaics in a wild plant. We went a little bit further with the lavenders. I've been working with lavender for 30 years now. We collected different parts, different modules. I call a module is a set of leaves and an inflorescence. We collect different modules in different parts of the plant and measure the amount of cytosine methylation of the genome. The genome-wide methylation is a quick and dirty measurement of epigenetic profile. Every graph corresponds to one different shrub, and the vertical axis is the percent of genomic cytosine that are methylated. Okay? First, we found that lavender shrubs are epigenetic mosaics. Different parts in the same plant have different level of methylation. So this is second evidence proving that plants can be epigenetic mosaic. But the interesting result was not this one, was that seed size variation within plants was significantly related to the level of methylation of the module. Or in other words, the large variance in seed size that you find in a lavender shrub and in many other trees, by the way, is closely related to differences in the level of methylation of the genome. Or, in other words, the epigenetic mosaics explain within plant variation in seed size in lavender. But it seems to me that the epigenetic mosaic hypothesis is insufficient to explain sub-individual variation, and things are a little bit more complex again. This is one aspect we are working on now, but I thought it would be interesting to tell you about this. It's in a stinking hellebore. Epigenetic marks are not perfectly transmitted from one generation to the next. They are partly reset from one generation to the next. Maybe it depends on the marker, but maybe 75% of reset or less. It doesn't matter. The interesting point was that we found that the level of mother to offspring transmission of epigenetic marks in stinking hellebore was related to seed size variation within plants. On the horizontal axis, you have the proportion of epigenetic loci that are faithfully transmitted from the mother to the offspring. And the vertical axis, you have the within plant variance in seed mats. Every symbol is a different plant. So those stinking hellebore plants with high proportion of loci transmit to the right, about 90% of loci transmit have their seed very, very similar in size. And if you go to the left, with plants which don't transmit the epigenetic mark very faithfully to the next generation, what you have is a very variable seeds in the plant. So my impression and my working hypothesis is that there is a complex connection between genetic feature of the individual, epigenetic mosaics, and some intervention, some effect from transmission of epigenetic mark to the next generation. I think there is still a long way to go before we understand what is actually happening. I have no idea whether the mechanisms are the same in trees, and long-lived trees or shrubs, annual plant and clonal plant. Probably in 10 years from now, we know that annual plants are working differently from long-lived woody plant or so on. But at least we are now starting to connect ecology with epigenetic, with genetic, sub-individual variation, and the functioning of the community. I'm going now to summarize and present you some working hypotheses. First thing is, we actually don't understand very well what are the causes of sub-individual variability. My hypothesis are individual genetic background is in some way affecting transmissibility of epigenetic marks, and also sub-individual variability in a direct way and through epigenetic mosaic. So altogether, working together is eventually producing what the degree of sub-individual variability we're seeing. On the other side, and the consequential domain, about the consequence, we have that, well, we know pretty well that sub-individual variability is important for interaction with seed predator or seed disperser or pollinator or so on. We know reasonably well that the sub-individual variability is important for the interaction with the physical environment, the light environment typically, the light grading in a forest, the light grading in the scrap of the hair layer. There is supporting evidence that all this together is impinging on fecundity, on fitness. We can only hypothesize now that fecundity will be affecting population dynamics, but only experiment will be able to demonstrate this connection. By the same token, differences in fecundity between individual and between population will have consequence for the community dynamic. So there is a lot of work to do. My takeaway message is this: a vindication of variance. For many years, ecology has been working with means, and very few times has worked with variance. I can understand, because 100 years ago, computing a mean with the computing ability, computing a mean was very easy, but not computing a variance. And Carl Pearson and other pioneers in biostatistics had to recruit women from the higher social classes in Britain to make the computation for the paper with the variance. There's the problem. Is we don't have any problem now to compute variance. The problem is not a problem of computing. The problem is a problem of sampling design and sampling intensity. Because if plants are so variable within them, we need a stronger sampling effort to sample plant well and to have a very good idea of the patterns of variation. Computing variance is no longer any problem, and there are reasons, there are conceptual reasons in ecology to vindicate the variance. So I think that's all. Thanks for your attention. I hope it was interesting. Thank you.
Thank you so much, Carlos, for this inspiring lecture that actually is opening us a lot of questions, and we receive a lot of questions through Twitter, and I was starting, and then...
Let me see, because I'm okay. A bit hair impaired.
Okay. If you look at the first, is stress triggering plant sub-individual variability?
Well, I think it depends on what do you mean by stress. If stress is the plant having to face an environmental gradient, for example, and water gradient, evapotranspiration gradient. In this case, the answer is yes, of course.
Thank you. Then we have another question here that deals again with some source of variability. How does the age or maturity of a plant affect sub-individual variability? It is sufficient to measure a plant once?
Good question. There are two components of sub-individual variation. What I call the instantaneous component is the variation you measure when you go to the field once and measure one day. And there is a long-term, a temporal variation. These two components, in theory, if you measure the same plant many times along the life of the plant, you can partition the sub-individual variation into an instantaneous component and sequential component. The data I have for maybe 6, 7 species for which I measured the same individual many times, and I found that the sequential component is relatively small in comparison with the instantaneous component. But probably it's not the same in all plant species.
Thank you so much. We have double technology now. So that's how does within plant trait variation compare to intraspecific trait variation of phylogenetically closely related plants?
Well, there's been, as far as I know, there's been only 2, 3 publications consider plant community, the different levels of variation in a plant community between species, between individuals of the same species, within individuals of the same species. Okay, so 3 levels. In this paper, they did a partition of total variation at these 3 levels. In some cases, not all, but in some cases, within individual variation was comparable to the sum of between individual plus between species variation. But I suspect that this probably will vary with the kind of trait you consider.
Thank you, and I will take just a last one that is, how do traditional morphological plant species concept hold against the high sub-individual variation?
Well, if by traditional we are talking about typological 19th century standards, I don't think this would be the nightmare of the taxonomy to include sub-individual variation. I don't know. I don't have a clear idea about that because I think the concept of species is at stake here. And the concept of species, you know, this is one of the most difficult terms. Maybe if you consider only morphology to define a species, what sub-individual variation introduces is a large amount of uncertainty. But if you consider the concept of species like in molecular term and in DNA sequence term, I would prefer that one. I don't think there is no problem, no contradiction at all. But I don't have a clear idea about that.
Yes, that is opening new question again. So thank you so much. But I will. I will now, on behalf of the Federation, to read the motivation for the Ernst Haeckel Prize Award to Professor Carlos Herrera for his distinguished scientific and academic career his profound knowledge of species and species interaction, his outstanding scientific contribution to deepen the understanding on the evolutionary ecology of plant-animal interaction, his energy and passion in transferring scientific ecological knowledge to knowledge users, promoting wiser conservation and management of natural resources, and raising public awareness on biodiversity and natural resource value for humanity, the Ecological European Federation confers to the 2017 Ernst Haeckel Award to Professor Carlos Herrera. And there is the Haeckel Prize medal